Preventing chemo brain with antioxidants targeting the spinal fluid. Preventing chemo brain with antioxidants targeting the spinal fluid. You're listening to Preventing Chemo Brain with Antioxidants Targeting the Spinal Fluid, presented by Hakeem, Ali-Bocas, Alexander on Wisdom for World Reading Club in association with UniquilibriuM. Today's cold reading is called Preventing Chemo Brain with Antioxidants Targeting the Spinal Fluid from neurosciencenews.com. Under the categories of brain cancer, featured, and neuroscience on September 13th, 2022. Summary. Using gene editing to boost the production of SOD3 in the choroid plexus reduced oxidative damage to brain tissue and cerebral spinal fluid in mice exposed to methotrexate. Source: Boston Children's Hospital. Up to 3/4 of patients receiving cancer chemotherapy suffer from chemobrain, a long-term side effect that makes it harder to remember things, maintain attention, and learn new information. When it strikes children, whose brains are still developing, their schoolwork and self-esteem may suffer. When we meet with parents and talk about the life-saving therapy we are proposing for their child's cancer, one of the most distressing potential side effects we sometimes describe is cognitive loss, says pediatric oncologist Lisa Diller, MD, Chief Medical Officer for the Dana-Farber Boston Children's Cancer and Blood Disorders Center. If we had preventative treatments that could be delivered during therapy or later in the survivorship phase of care, that would be game-changing. In a study published in the journal Neuron on September 6, collaborators Maria Leitinen, PhD, and Nema Konarek, PhD, in the Department of Pathology at Boston Children's Hospital take a first step in that direction. Their vision: treating the cerebrospinal fluid that bathes the brain and spinal cord to protect the brain from chemotherapy's toxic effects. If we can treat the CSF, which is easy to target, we can potentially treat the brain itself, says Karanek. Investigating chemo's effects on the brain. Leitinen has conducted pioneering studies showing the importance of the CSF in promoting brain health and growth, together with the choroid plexus, the little-known brain tissue that produces CSF. Separately, Konarek has been studying the metabolic effects of the chemotherapy drug methotrexate on cancer cells. Methotrexate is used in children with acute lymphoblastic leukemia that has spread to the brain, as well as osteosarcoma and certain tumors of the central nervous system. It is also used in adults to treat leukemia, lymphoma, breast cancer, and lung cancer. The two labs teamed up. In an initial study of mice, they found that exposure to methotrexate caused oxidative damage, a metabolic imbalance leading to the production of toxic oxygen molecules to both the CSF and the choroid plexus. Nerve cells in the hippocampus, the brain's main center of learning and memory, were also damaged. And in behavioral testing, the mice showed increased anxiety and impairment on tasks involving short-term learning and memory. Using advanced laboratory tools, Leitner, Kanarek, and colleagues further showed that methotrexate prevented the choroid plexus from secreting a key enzyme, superoxide dismutase 3, SOD3, into the CSF. Superoxide dismutase 3 is a natural antioxidant that normally protects cells in the brain and other tissues by helping break down potentially toxic molecules. The researchers think that without SOD3 delivered by the CSF, brain cells are more susceptible to damage, reversing toxic brain effects of methotrexate. Do the same effects happen in humans? The researchers first studied human neurons generated from stem cells. When they exposed the neurons to methotrexate, they produced less SOD and showed evidence of oxidative damage. They also studied CSF in samples from 11 adult cancer patients who had received methotrexate chemotherapy for lymphomas affecting their central nervous system. Compared with 12 cancer-free controls who did not receive methotrexate, these patients had reduced levels of SOD3 in their CSF and an increase in markers of oxidative damage. So could adding SOD3 back protect the brain? Using a gene therapy approach in the mice, the research team boosted SOD3 production in the choroid plexus. When they gave the mice methotrexate, the animals' CSF and brain tissues were largely free of oxidative damage, and there was less evidence of anxiety and memory impairment. With more work in the future, these findings have the potential to prevent or treat one of the most concerning late effects of methotrexate, a cancer drug we use frequently, says Diller, who was not part of the study. Exploring treatment possibilities. Leitner and Kanarek now want to explore the effects of other chemotherapy drugs on the CSF and choroid plexus. They also want to better understand how methotrexate and antioxidants affect other parts of the brain and test the efficacy of antioxidants given directly rather than through gene therapy in a living chemo brain. Antioxidant treatments could potentially come in the form of an IV injection, a nasal spray, or an that's interesting. Restarting. Antioxidant treatments could potentially come in the form of an IV injection, a nasal spray, or an intrathecal injection through a spinal tap directly into the CSF. Such treatments could possibly be given together with the chemotherapy itself. The most immediate opportunity might be in patients who are receiving intrathecal methotrexate for brain cancer or for leukemia or lymphoma with brain involvement, says Kanarek. Another potential approach, yet to be tested, is simply providing antioxidants through antioxidant-rich foods or dietary supplements. This first study is just the tip of the iceberg, says Leitner. If we could correct the collateral damage of chemotherapy and make even a small improvement in the trajectory of patients' lives, that would be really exciting. Abram Jank, DPH, in the Leitner lab was first author on the paper. Neuroscience News asks you to see also, under the categories of featured and neuroscience, on August 4th, 2022, your brain is a prediction machine that is always active. Neuroscience News continues. with funding information from this article. The study was supported by the American Heart Association Pre-Doctoral Fellowship Program, the National Institute of General Medicine Sciences, the National Research Foundation of Korea, Boston Children's Hospital, Gabriel's Angel Foundation for Cancer Research, STARR Cancer Consortium, the National Cancer Institute, the Harvard Stem Cell Institute, the National Institute of Neurological Disorders and Stroke, the New York Stem Cell Foundation, and the Intellectual and Developmental Disabilities Research Center. Okay, excuse me for that. Disclosure. Maria Leitinen and Abram Jang are co-inventors on a provisional patent application related to this paper. About this chemotherapy and neuroscience research news. Author Bethany Tripp. Source Boston Children's Hospital. Contact Bethany Tripp, Boston Children's Hospital. The image in this article is in the public domain. Original research. Closed access. Choroid plexus CSF targeted antioxidant therapy protects the brain from toxicity of cancer chemotherapy by Avram Jang and others. Was I saying Abram Jang before? It's Avram Jang. No, it's my bad. All right. Neuroscience news continues. Yes, neuroscience news continues with an abstract from Choroid plexus CSF targeted antioxidant therapy protects the brain from toxicity of cancer chemotherapy. Highlights. MTX induces oxidative stress in mouse choroid plexus, CHP, and CSF. MTX damages human neurons and diminishes antioxidative capacity of patient CSF. CHP targeted SOD3 gene therapy replenishes protective capacity of mouse CSF. SOD3 gene therapy prevents MTX-induced lipid damage, anxiety, and memory deficits. Summary. For many cancer patients, chemotherapy produces untreatable lifelong neurologic effects termed chemotherapy-related cognitive impairment, CRCI. We discovered that the chemotherapy methotrexate, MTX, adversely affects oxidative metabolism of non-cancerous choroid plexus, CHP cells, and the cerebrospinal fluid, CSF. We used a choroid plexus targeted adeno-associated viral vector approach in mice to augment CSF levels of the secreted antioxidant SOD3. Wow. Adeno-associated viral vector SOD3 gene therapy increased oxidative defense capacity of the CSF and prevented MTX, that's methotrexate, induced lipid peroxidation in the hippocampus. Furthermore, this gene therapy prevented anxiety and deficits in short-term learning and memory caused by MTX. MTX induced oxidative damage to cultured human cortical neurons, and analysis of CSF samples from MTX-treated lymphoma patients demonstrated that MTX diminishes antioxidant capacity of patient cerebrospinal fluid. Collectively, our findings motivate the advancement of CHP and CSF targeted and antioxidative prophylactic measures to relieve CRCI. Neuroscience News asks you to join our newsletter. Sign up to receive our recent neuroscience headlines and summaries sent to your email once a day, totally free. We hate spam and only use your email to contact you about newsletters. You can cancel your subscription anytime. Neuroscience News posts science research news from labs, universities, hospitals, and news departments around the world. Science articles can cover neuroscience, psychology, AI, robotics, neurology, brain cancer, mental health, machine learning, autism, Parkinson's, Alzheimer's, brain research, depression, and other topics related to cognitive sciences. You've been listening to Preventing Chemo Brain with Antioxidants Targeting the Spinal Fluid, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM.